So, thank you. As you have just heard and seen, this meeting started to be recorded. So, we don't have questions or comments, maybe at the end of the session. So please, Professor Horaf, if you would like to share your screen and start your presentation.
Yeah, thank you very much, and thanks for the nice introduction. Can you see the screen now in the presentation mode?
Yes, yes.
Oh, well, then we can start. Yeah, thanks again for the invitation and for coming here virtually to the CEF. My talk within this series is sort of bringing together a lot of the projects that we do in the institute here of medical microbiology, parasitology also, where it is about new drugs against neglected tropical diseases. Now, neglected tropical diseases, I have to see how I've moved that forward, sorry. I'm fighting here. Okay. Neglected tropical diseases are actually a sort of restricted list of diseases restricted by the WHO in this list. I'm sorry, I'm still struggling with getting the steering instrument here. Sorry. Of about 20 NTDs, and this is the only slide I have to say that is in German, because the outline in this green color is very nice, and it is from our website, from the German network that that has already been mentioned in the introduction. That is a sort of a think tank and advocacy network for the fight against tropical diseases in Germany. And as you see, typically in the tropical areas of all the continents, here is shown Africa, you have a concentration of the tropical neglected diseases that, of course, the more you get towards the equator, the more countries you have with several of these diseases. My focus of interest and the focus of interest of the institute working on parasitology is mainly the filarial infections. They are very frequent infections with more than 100 million infected people worldwide, and they are usually transmitted by insects. So in the case of the lymphatic filariasis, that leads to disfiguring limbs. This is why it has the name lymphatic filariasis. There are mosquitoes, sometimes identical to the malaria transmitting mosquitoes, therefore you have similar habitats. Whereas the other big disease we are working on, Onchocerciasis, transmitted by or induced by the worm Onchocerca volvulus and transmitted by simulium flies, is bound because of their breeding sites close to rivers and is therefore also called river blindness. In common is that both diseases, both worm species, have a long life of 10 to 15 years. In case of lymphatic filariasis, these adult worms reside in the lymphatic tissue, and then they give birth after sexually mating to larvae, to microfilaria that appear in the blood in the case of lymphatic filariasis, and mainly in the skin in the case of Onchocerciasis. Here the location of the adult worms is also different. It is in subcutaneous worm nodules, and we'll see a few more images in a second. So the important to know is there are long-lived adult worms and rather short-lived larval stages that are transmitted from the mosquito into the human host and then after mating back to the transmitting insect. So the disfigurement and disease is different with lymphatic filariasis because the lymphs are chronically infected. You have changes of the lymph flow that may lead to genital disfigurement like hydrocele, so it's a water-filled human scrotum, and here you have elephantiasis that can progress from very small to very large. In onchocerciasis, the name river blindness is due to the fact that some of the larvae, the microfilaria, do not only migrate to the skin, but also to the anterior chamber of the eye. And through chronic infection and inflammation again, there will be scarring and in the end blindness. And like 20 years ago in Africa, this was a severe impact on the daily adjusted life years. A lot of men in the working age in farming were taken out of their jobs because of blindness. They couldn't really look for their living and death of their families. This is now improved, but we still have a lot of millions of cases throughout Africa. Now, immunology does a lot to the shape of the diseases. Usually the parasites manage to keep the immune system at bay and downregulated. So for example, in this case, the normal appearance of onchocerciasis, you have a worm nest here and here in the thoracic cavity. But you have to imagine that the skin will receive millions of little larvae, the microfilaria, and you may have in a cubic millimeter of skin, you may have up to 100 larvae. So you can imagine how many larvae the body will contain, and still you don't see a lot of inflammation. And this is due to the fact that the parasite actively downregulates the immune system and has a lot of implications, I think, even into COVID-19 these days, because the parasitic infected people usually have a downregulated immune system that does definitely not overreact as we know it from people without worms. Overreaction occurs in some rare cases, and here you see what happens. The parasites, the larvae, are actually being killed, but so is the skin, so to say. There is strong dermatitis, and the patients suffer much more than these patients over there with the hyperreactive form. So these onchocerca nodules are usually easily to be palpated during examination, and they are motile. And I have brought a little movie to underscore that. Hopefully it works. Yeah, it does. So you can really move this nodule, and since it is not painful, patients often don't know, particularly if they are on the back and nobody tells them, hey, you have something here, and they are absolutely happy. And you see this person also has not much skin affection, so belongs to the immunologically silent hyporeactive form. Now you can operate those nodules as a therapeutic intervention and then digest the human tissue around it. And then you see how many adult worms of up to 70 centimeters of length a single nodule may hold. This has been our record nodules over 20 years. Usually you have only 2 to 3 female worms, and the males are much smaller, and they are often not present in the worms because they are on migration. Now the skin, you do a so-called skin snip, what you see here on this video. This is strictly intradermal, not subcutaneous, because you don't want to pick up blood vessels, and then you have to put them into a saline solution or PBS buffer in order to allow for the little microfilaria to evade the tissue, and then you can count them in the same plate here. So now you may know that these diseases are targeted by WHO. We come to that later. For elimination. So you need to have tools to do that. And the most used tool, apart from some vector control and mosquito spraying activities, particularly in the past, the actual intervention tools are drugs. But the drugs, there are 3 for lymphatic filariasis and basically only 1 that really works for onchocerciasis, ivermectin. They mainly attack the larvae and lead to the fact that there is no transmission for a certain amount of time. But the adult worms sitting here and here, they continue reproducing. So after a few months, the skin in onchocerciasis or the blood in lymphatic filariasis will have been repopulated. And so transmission is ongoing. Therefore, you have to repeat treatment very often. And currently, this is done by annual or biannual mass drug administrations in endemic areas. So you don't even examine every patient, you just do an epidemiological survey. And if the disease prevalence is more than a certain percentage, usually 2%, then you start interventions. And you can see already from the figure that if you are at a 1-digit percentage prevalence, you'll treat 90% of the people without a reason. This is only due to the fact, only possible, ethically possible, because these drugs have almost no adverse reactions, and therefore you can do mass drug administration without diagnosing. But we will move away from this in the next time. I will come to that when I explain the new WHO roadmap. Now, almost 10 years, 8 years ago now, in what was called later the London Declaration, drug makers and WHO plus donors like Bill Gates, see him here, convened and agreed upon to donate a lot of the drugs for free to WHO according to their needs, because it was accepted that the people suffering from the so-called neglected tropical diseases would not have the means, financial means, to pay for their health, nor did the governments in endemic countries. And so this program was launched in 2012 with the, at that time, very optimistic and challenging roadmap to eradicate one disease, the Guinea one, eliminate 4 others. Elimination in this context means regional or continental elimination, so eliminate it from Latin America also, and control 5 more. And if we go to elimination and eradication, you will quickly see, if you're into this field, that all these goals have not yet been met by 2020, which was the original deadline. Nevertheless, it was an unprecedented agreement and building of a consortium of a lot of big pharma plus donors. Notably, Germany at that time was not part of it, and one of our ambitions of the German network is that for follow-up conferences like this, Germany may take a more active role. I'll come to that. So how does mass drug administration work? Basically, it is the challenge of reaching the unreached, because the drugs are there and they need to be now distributed and in a meaningful way. So with a high enough coverage. If you only treat 10% of the people in need, you won't get anywhere with regard to disease control. You need at least to be in the figures, and there are exact modelings about that we don't cover today, in the range of 80% of all people who are eligible. In 2016, and there have been similar data for the following years, there have every year been record-breaking commitments of the pharmaceutical industry in the range of several billions of drugs distributed and donated first to the WHO and then also distributed. And distribution works as follows. The countries, through their ministries of health, tell the WHO how many people in need there are and get shipments of lots of cartons of drugs that then will be distributed according to the lists of the respective districts and regions, then distributed to like peripheral structures like a district hospital, and then distributed in mass drug administration. And currently there is a big process ongoing to digitalize all these structures so that you don't have to come up with a lot of time delay, like here going through lists and realizing, oh, I have fallen short of maybe 1 million drugs in my country. This has been identified as a major roadblock to better performance. Now, how do mass drug administrations in the communities work? It's done under the responsibility of the community itself through community health volunteers. And of course, according to very simplified drug regimens, for example, for ivermectin in onchocerciasis, there is body weight dependent amount of drugs that you have to take, and the body weight through body mass indexing is of course translated into height, and every little village you can find a mass drug administration pole that is kept in the district hospital, and it's of course normed, and here you can directly read how many drugs this little girl, for example, has to take. And there's also exclusion criteria. So if you are not able to get to the opposite ear with your hand, if you're not yet able or no longer able, then you are not eligible for the drug treatment. Now, these are the current strategies, and let's see how that works. The WHO is bringing together now more accountability. This is a big buzzword of the roadmap that only by last week has actually been agreed upon and is now sort of law for the WHO. So accountability is very important, and since a couple of years they are doing it and really say, okay, there are countries that have a good geographic coverage or a less than 100% coverage, and they do statistics over the 4 years, and you see the long list of countries that can still improve. There's only a few green countries that do it in an ideal way. And this has led to several modeling studies by epidemiologists, like this one from 2015, where it's been said that despite the ambitious goals of eliminating some diseases by 2020, and now there is a new roadmap saying a certain percentage of countries should have eliminated onchocerciasis, for example. There will still be remaining mainly in Central Africa countries where there is no clear end in sight. So 2045 plus just means the confidence intervals end there, and it may also be taking until 2060 or so. So my team has been active in Africa for a longer time, and we've done mainly clinical trials, to which I come later. That gives us a lot of data from randomized trials, so they are well controlled. And therefore I was asked to donate or to feed this data into modeling systems of mainly the Rotterdam group of epidemiologists that do a lot for onchocerciasis, elimination, simulation models. And they did a high number of simulations. And the question is, after so and so many rounds of ivermectin, where the microfilaria go down, then after a while go up, back again, go down, go up. If you do that, say, for 10 years, what is the chance of the onchocerciasis, the remaining onchocerciasis, to die down on its own? And what is the chance of a resurgence? And you see in 15% of cases, there will be a resurgence. And it's of course dependent on a lot of factors I cannot go into. It's very complex. But we have the problem that by the mass drug treatment with a drug that kills only the worm larvae, we have to repeat the whole thing very often at high coverage, and still we may not end up where we want to go. And particularly if there are some hiccups, like the current COVID-19 crisis has, for example, led to an interruption of mass drug administration. And there are already modulations that say if you interrupt for 1 year, you fall back by 2 to 3 years. So there may be more than this corona crisis coming. Anyway, one thing is support. And here you see a lot of the major donors. You see there are not so many Europeans around here. You see that there is a lot coming from the Arab nations, particularly the oil-rich countries, and you know this gentleman here, Gates Foundation, and they really get out a lot of funds that are also necessary. As I said, the German network has sort of lobbied the German government also to start in the extra donation round, in addition to what they already give to WHO, and for the launch of the Global Health Program by Minister Spahn last year. He announced that they will now start supporting one part of the WHO neglected disease, tropical disease department. That's a special program for to eliminate NTDs. And they start funding on that. And we hope that in the next summit that follows the London summit will be hopefully a Kigali summit that has already been planned, as you see, for this year, but was cancelled due to coronavirus, that it will take part next year, and there will be a commitment by the German government as well. Now, a very major and important event has taken place only last week. You can see it here. And you can see the main protagonists, of course, the General Secretary and the director of the NTD program, a Tanzanian researcher with whom we have already done several projects together. She's now at the WHO, and they have done a new, they've produced a new roadmap of about more than 100 pages with a lot of annexes, and you go really down in the nitty-gritty details. You do, they did a survey of what is necessary, so diagnostics are very necessary, they are in the red circle, and also medium-term necessity is, but in the end it will really help and shift the paradigm, is to develop a new drug that is active against the adult worms. And such a drug is called a macrofilaricidal drug. And now, in response to, or even before that, there have been already consortia funded by the Gates Foundation and others to develop this. And one way to do, where we have already produced prototype drug in doxycycline, is a mode of action that is really unique to the filarial worms and to most of the human pathogenic species. And this is that they contain endosymbionts, so endobacteria that never do infections. They are living like mitochondria, always inside the cells. Here they are stained with an antibody against the surface protein to these bacteria. So they live in a worm. Here's a worm section. This is the cuticle of the worm. Here is the outer wall of the worm, the so-called hypodermis. And this is a female worm with two branches of uterus with little babies here, the morulae. They are still in embryonic stage. Here are the oocytes, and they will further develop. So in all these newly developing embryos and in several parts of the adult worm, there are these Wolbachia endosymbionts. They are related to Rickettsia, and so the people fit in medicine or pharmacy of you, they will know that the Rickettsia are good targets for doxycycline. And this is what my team and I thought back 20 years ago in Hamburg when we first detected these Wolbachia and said we should try doxycycline. So these is an image of the different development stages of this embryo. So this is the egg, this is the fertilized egg, this is dividing into a so-called morula, and then a little worm larvae develops within an eggshell here and then becoming a free microfilaria. And you can see that in histological sections of these adult worm nodes. So you see the embryos again, you see some eggs somewhere dispersed in there. You see these pretzel stages. They look like a pretzel, this is why they are called so. And here the ready to go microfilaria, they will leave the female worm, live birth, and then they have to migrate through the nodular tissue further up to the skin or to the eye. Now, knowing that from animal models that we did back in Hamburg, where I worked before Bonn, that doxycycline works against filarial worms, we were quickly invited by Ghanaian colleagues to go to an area between the coastal section and the savanna north. Here there is still rainforest area. It's where the cocoa growers live, and where you really have a good example of how hard it is to hit the last mile. We went into the villages and started a doxycycline trial, of course controlled, and then after a while of doxycycline, the patients had the chance to get their nodules aseptically removed. And they were then taken under slide producing apparatus to produce histological slides and analyze them. And what came out really crystal clear is the patients who had not received doxycycline still had their Wolbachia endosymbionts, and the worms were fertile as ever. However, patients that had received doxycycline for 6 weeks of treatment, and this is the issue that we have to come back in a few minutes, it's a long time, but after that treatment time, all the Wolbachia that you see here are gone in the treatment group, and you also see that the embryo, that the uteri are no longer full of embryonic developments, they are empty. So these worms have become sterile, and this sterility holds on for a long time, at least 2 years, probably longer, probably a lifetime. And the worms don't live any longer for 10 to 15 years. They usually die within 1 year to 18 months. So it is a slow macrofilaricidal effect, which is good because a quick macrofilaricidal effect would lead to strong anaphylactic reactions because of the big tissue that falls apart. So this is actually what you want. So doxycycline is a good prototype drug and is used already. We did a lot of clinical trials. This is where we get our samples from, reducing the time to 5 and 4 weeks, trying out whether this works also in areas where there has been ivermectin resistance already. Then we went to another part of Africa, to Cameroon, to find out whether it works there as well. And here we even did a community trial where a lot of patients were treated with a high compliance and with very good results. So basically this drug can be used, but of course it has the shortcomings that children under the age of 8 are not allowed to be given the drug unless there is an emergency, because that may lead to disturbances of teeth and bone development. And of course you must not give it to pregnant women. So a big proportion of the population in African communities are then excluded. And of course we want to overcome that by new drugs. I come to that in a short time. Yeah, this just is done modeling with another modeling group showing that the different treatment regimens are all basically the same. I go over that. And with this data and a lot of administrative preparation taking a few years, the program for elimination of onchocerciasis in the Americas, which is now left to only 2 countries, namely Venezuela and Brazil, here the red dots, want to end the filariasis control quicker than with ivermectin by also distributing doxycycline. So I'm happy that they finally decided for it. It takes time with WHO, we all know that. We have seen the criticism of some people due to WHO in the COVID crisis, but WHO has their mechanisms. They cannot just decide. They have really SOPs how decisions have to be done, and therefore it may take longer than you want sometimes. But now they are on their way, and we hope that the Americas can eliminate onchocerciasis altogether. Now, lymphatic filariasis, you have seen the disfigurement. In Ghana, for example, it's distributed along the coast, along fishermen's villages, because the Anopheles mosquitoes that transmit them are to be found in breakwater. It is just behind the coast. There is already salt and sweet water mixed. How do you detect filaria here? You use an ultrasound instrument. Since there is no electric power, you have to do it by battery powering. And in the scrotum of males, you can see the worms moving. This is here on this image. This is a scrotum. You can see where the transducer is held. And here there are enlarged lymphatic vessels, and you see the worms moving. Now, we also did a lot of studies that I shortened because it's already some time ago. And basically we developed regimens that also have entered their way into the major textbooks of tropical medicine. And we have done over the last years additional studies to reduce the daily dose. And this is what we can do basically for doxycycline with one addition that I'm going to talk about now. And this is the problem with treatment of lymphatic filariasis is that you can treat the infection with other drugs, ivermectin and so on that I showed you, but you cannot do a lot with the disease, because when disease has already progressed, there is scarring of tissue and a lot of other disfigurement, so you cannot go back on this just by wiping out the infection. Actually, in these people, because they're also sort of overreacting to the worms, there are usually, after a while, no more worms present, but the disease progresses because between the toes, people usually have little fissures, and exogenous bacteria from the road or from a rice field or whatsoever enter the bloodstream, and they cannot be transported away by the lymph because the lymph is dysfunctional because of lymphatic filariasis. And then they progress, and in the end they can hardly move. So what we found during some of our trials was that the patients with lymphedema would not progress. So here you see the stages of progression, and here you can see that this progression is not happening when people are under doxycycline, but when they are under placebo. So we did another study specifically for lymphedema, and this was confirmed. But this is not enough for a treatment recommendation by WHO yet. For that, you need to have more confirmatory studies and also from more continents. And this is what we are now doing within the funding scheme of the BMBF, in their funding scheme Research Networks for Health Innovations in Sub-Saharan Africa. And there are 5 networks covering these countries, and ours is this one here with filariasis. And one of the major aspects that we are doing in these 3 countries is trying to improve this situation with lymphedema patients. And it can be improved, as we have shown, from already progress stages back to almost normal when the patients have received doxycycline. So this trial will end next, or has already ended, is now, we are 1 month away from the database lock. So when you really lock and close the database in a blinded manner, and then you are only allowed to deblind. So we hope that we can confirm that. This was, by the way, also a very nice international exercise, because we were joining forces with USAID funded groups from the United States, active in India, in Sri Lanka, and in Mali. And so we have a lot of countries, 5 RCTs, 5 randomized clinical trials to confirm what we have found before, and we hope that this will work. It's already a very complex thing to get all these 5 centers with different countries, different ethical regulations into all on board and define a multicentric protocol with about 10,000 entries for every patient during the study. So you need a study management team. And Ute Klarmann in my group has set this up over the years and was really instrumental in putting this protocol together. And of course, in Africa, the times where you just went to Africa and did a study are long gone. African states also have their FDA, so Food and Drugs Authority, it's called, in Ghana, and there are similar equivalent authorities in other countries. So you have to, of course, to submit any drug trial. And this is a huge effort. So the protocol looks like this. So it's like a book with these 10,000 entries. And there have been 3 committees, university committees and national committees looking over these studies before they are approved. And so I'm very grateful to my major African partners, Alex and Linda Debra, for getting this through in our African partner country, Ghana, at the Kumasi Center of Collaborative Research. This is just an image of patient recruitment. And this is really necessary and also recognized by WHO that that they have to really be aligned, their goals, with sustainable development goals. And one of, in the part 3, good health and well-being, is of course also universal coverage and access to drugs. So the MDA, Mass Drug Administration, that does not care about disease and morbidity is no longer possible. They also must care for morbidity, and therefore we hope we can deliver a tool with doxycycline. Now, going back to development, new macrofilary sites, just a few more slides. That has been a long endeavor, more than 10 years. It was originally funded by the Bill Gates Foundation, and they were going back and forth whether they should fund new drug development because they are very much into technology, and they wanted to find out first whether, when looking for a new anti-helminthic, do we know the mechanism of action? And the answer for all drugs that deplete the Wolbachia is, of course, yes, we know you need to deplete the Wolbachia by 2 logs, and then you have your antiparasitic desired effect of warm sterility and macrophyllaricidal effect. And we can mimic this in a high throughput manner using insect cell lines that also contain close relatives of the warm Wolbachia, this insect Wolbachia, and you can see there is a difference between normal status and cure that can be used to set up high throughput screens with more than 10,000 compounds per month. And so the Gates Foundation formed a consortium, which was worldwide and contained different labs, also in Asia. And there was a business plan. And I'm just showing that just to make you aware of how much attrition there is. So you start with almost 500,000 of compounds. And then there are some hits. This is already a strong reduction. And then you want to do maybe some chemical modification to optimize your lead. And then you go into animal models, and that always cuts it down. So in the end, you have a few, if any, new drugs. And actually, we were lucky. We did find one drug out of all this endeavor. And others came through a few other chance type of events that were not really planned. A lot of drug companies gave their compounds. This was all organized, as I said, by the Bill and Melinda Gates Foundation, plus the Drugs for Neglected Disease Initiative in Geneva, with a lot of partners. And one of the candidates now in the clinic is a drug called Tylermec, or AbbVie 4083, that was found in this in these preclinical experiments. And it comes from a mother drug that was actually used for better cattle growing, because it's an oral only antibiotic. So you change the cattle's microbiome and then they grow faster. This is no longer used as such, but by changing here some of the doing some modifications here, the chemists at AbbVie could find more potent activity of a second generation tylosin, analog 200-fold more potent. And so we went, the drug went into our helminth filaria model. It's a helminth filaria called Litmus sigmodontist that can do a cycle in mice, and transmitting vector is a mite, so not an insect, but a spider, so to say, an arachnoid. And there can be mice and cotton rats that are infected. And so you can do your in vivo treatments here. And this is a busy slide. Maybe I show you the microfilaria, the larval reduction over time, by the new compound AbbVie 4083 in comparison to the standard drug doxycycline, for which we know it's not enough if you treat it for 14 days. And you see the microfilaria are not gone. So the microfilaria here are gone because the embryos are being depleted. You can see that in that slide, and also because the worms are being killed. And this has now made it into a clinical development portfolio by DNDi. You can actually go to their website and then you see the same pages. There are some candidates, and I just pick out the Tylermec or AbbVie 4083, where we are also now new partners for the clinical development. And there is another one, amodepside, that is already known in veterinary medicine, and Bayer wants also to donate it for humans. And there is another one I cannot talk now due to time constraints. It's called oxfendazole. It may be also developed against cysticercosis and other tapeworms, but will probably do a very good job also for filariasis. Now, when you do clinical trials, you have a lot of hundreds to thousands of patients, and every patient's got several onchocerciasis nodules, like the ones you've seen in the beginning. So the histologist having to look at all that, it will take ages to really assess. And even if you share the work and have 2 or 3 experts, it will take a long time. So the Gates Foundation was happy that we made them the suggestion of using knowledge on artificial intelligence that was available at the company Capgemini. They are based with one of their many bases in Cologne, to work together and have artificial intelligence apply on reading of the nodules. And you see such a nodule has a lot of warm sections that the computer must detect first, and then must see what's in there. You may already see, okay, here there are some embryos, but of course it's not as easy as it seems. Very often the pictures are a bit ambiguous, so a lot of training is necessary for the computer to know where to look and to know how to assess. But once it's done, and we have already trained, I don't know, several thousand sections, and arrive at a very good evaluation accuracy. So 97%, for example, regarding vitality or presence of Wolbachia in comparison to 2 different experts that have aligned themselves already. And this will be a time saver. And as you know, the WHO roadmap is now only for 2030. We have, of course, to save time. So phase 2 now really needs to start and has started. And phase 3 will be sort of telescoped, like in the current COVID vaccine trials, into phase 2, so that at some decision point we already decide which of several doses we test in phase 2 will then make it into a phase 3. And hopefully by the mid of this decade, 2025-26, there will be a new drug ready based on this principle of the Wolbachia. Now, in Bonn and Cologne, in the introduction, it was mentioned, we have a site of the German Center for Infectious Disease Research, or Deutsches Zentrum für Infektionsforschung. And one of the major activities is to define new antibiotic drugs. And we have found one that we're going to develop, which is also active against the Wolbachia. It works similar to rifampicin, but has some advantages, which we can maybe discuss later. I want to come to a close. It's produced by a soil bacterium and has been recloned in a better to handle bacterium. There are like 20, 30 steps of synthesis, and all these synthesis genes had to be recloned in a different organism. All this works now. And what it shows is, when applied in our mouse and rodent models, is that the corallopironin can actually bring down the worms in these rodents in only 7 days to almost 0, and the microfilaria are all gone. So we anticipate that humans later on would have to take between 100 and 200 milligrams per day. So far, there are no toxic effects. And the nice thing is we can develop this antibiotic also for other diseases like gonorrhea and chlamydia. I just jump over that a little faster in order to come to an end. Chlamydia, you see here nice immunostaining slides. It shows that it is actually better than rifampicin at a lower dose, because it has a very good tissue penetration activity. This is very good. It also penetrates in staphylococcal abscesses. And this is the second way how DEZIF wants to bring this project forward. And DEZIF's mandate is not to do only basic research, but really to accompany development through the sometimes a bit boring safety requirements, safety controls, like does it target other organs? How is it excreted? Is there genotoxicity? This is all a very standard checklist that you have to work off. It's not so much for researchers, but you have to do it. And if we succeed and are in the clinic in 3 years, then it will be one of the first really publicly funded antibiotics. The original reason to fund it was because it's so nicely active against the Wolbachia and good for filaria, but we now develop it also against other bacterial infections. And one really crucial step towards it has been fulfilled actually today. It was a signing of a contract to a Belgian company. You see some prints here that where we can source out the production process. So now it will get into the hands of industry, still funded publicly, but eventually this will take over. Takes, of course, you may imagine, a lot of time to get the patent rights, to get all the legal stuff right. It took us several months. And a lot of DEZIF consultant help, but now we can give it further. And we need, of course, still to do some toxicity study, particularly the final ones when we have already GMP, good manufacturing practice equivalent gradients, purity of the drug that is then possible to apply for clinical phase 1. And I think with all that, I'm at the end. Just to start with the latest project, this is the team throughout DEZIF and I think 5 or 6 sites in Germany where we do collaborate about the development of this antibiotic, which holds a few patents and has won a few prizes, also in the biotech area, and comprises a lot of people in Bonn and at the Helmholtz Center for Infection Research and the DEZIF headquarters, among others. And of course, clinical trials and preclinical trials would not be possible without many teams in the country. So the preclinical development part is done by Professor Mark Hübner, who now holds a DEZIF professorship, and his team, and my, some of my Tanzanian and Cameroonian and some Ghanaian partners at a meeting. And this is our major collaborating institute, the Kumasi Center of Collaborative Research in Kumasi in Ghana. And I told you about the 10,000 data per one patient in the study, and you need a few database experts, and they are here on this image, also the artificial intelligence. And this lady is managing the coralloperonine project. She just sits here. So I'm sure I have forgotten maybe 1 or 2 people, hopefully not in the audience. So if you're not on this list, I'm sorry, but you all, of course, belong to the team. We got a lot of funding, and otherwise these projects would not be possible. And of course, I think it also gets into the fields of applied topical research that is really constitutes a close link to the Central Institute for Entwicklungsforschung, to the CEF. Here I'm at the end, and thank you for your attention, and maybe you have a few questions that we can then discuss.
Yeah, thank you very much, Professor Horaf. This has been really a very impressive presentation, I have to say, not only because of the effects of the parasites on the human body, but particularly because of all the efforts that one can see are made globally, and particularly by your team. So please, if you have questions, comments, just raise your blue hand or open your video. Just to remind you that this session is being recorded, so if you don't want to be seen, you just can write your comment or question in the box here, in the chat box. I don't see any hand here, so I will take the opportunity to ask a question that I have. And coming from the social sciences, I would like to ask you, Professor Horaf, what is a bit the history of this lymphatic filariasis? Because I can imagine it is really for a long time where it is, and how people have managed till now, maybe with some traditional knowledge, or people just said, okay, we are inhabilitated forever, or they have really some kind of treatment. And yeah, my second question has also to do a bit with this history of this disease, and it is if there are some environmental factors that favor the spread of this disease, or the, let's say, the constraints this disease, just to see a bit of history of how this has been treated. And
thank you very much. This is very important questions. And so I hope I can recall them all. Maybe to start with the second one, habitats and where they come from. So every area where Anopheles mosquitoes in Africa, or even Culex mosquitoes like to breed, is potentially a breeding site and a transmission focus. In Africa, it's very much overlapping to malaria areas. Therefore, there have already been ideas and attempts to do joint programming with vector control. In areas like India, for example, you also have urban lymphatic filariasis, also in East Africa. Here, the transmitting vector is not Anopheles, but Culex. And as you may know, Culex breeds even in wastewater. And therefore, you know, if you have open wastewater canals in some mega cities in developing countries, these are breeding sites, and all of a sudden there may pop up lymphatic filariasis breeding. Otherwise, it's just some water wells that are unprotected. And basically, with regard to malaria control, the same, with regard to Anopheles control, the same as for malaria control applies. So a factor that I have seen, I've also read, but not so much about, is, of course, when you, for example, change from growing some fruits that are in dry soil, you change to growing rice. And you have a lot of basins where Anopheles breed. This is definitely a factor. And the second factor with rice growing is that once you have developed an elephantiasis, your skin is very much under tension, and you have these little fissures between the toes and also between the hands here, and the fingers. And there can exogenous bacteria can enter there, and particularly if you are a rice grower standing all day in the mud and you grow your rice plants, of course this is a risk factor. And I have heard very often from a lymphatic filariasis area where we worked in Tanzania that people have stopped being rice farmers and have changed to, say, cocoa or other crops that they have to work with, because otherwise their feet would still grow larger and larger, and this helps. So with regard to the elephantiasis or the thick legs, the exogenous bacteria are a major factor. And this is where also traditional medicine comes in, because there are very good recipes of antiseptic plants that people can put on their feet to get the fissures closed and prevent entry of bacteria. I think I am just looking quickly whether on one of the slides there's actually a pad where this has been tried to work. Let me just see internally. Oh no, there's nothing, because I have a picture of a traditional healing with some herbs. So it's a green paste that is brought over an ulcer, and this helps, of course. So I think that was what the people did for a long time. What we realized when we first entered lymphatic filariasis endemic areas is that the people don't really know what the reasons of transmission are, because the majority of people doesn't even see symptoms. They are completely normal. They just have the little larvae swimming in their blood. There may be 1,000 per milliliter or even 10,000. And because of the immunosuppression, people don't know it. So they only see the very few overreacting ones with elephantiasis, and they stigmatize them. So they say, you must have done something wrong, and this is why you now get the sick food. And we had to do a lot of advocacy first and explaining that it is mosquitoes that actually transmit the disease. So I know the word for mosquito in several of the native languages, because I had to explain that. And you could always feel that many people say, aha, okay, this is then a completely different thing. And so it is not your individual's fault why you get infected or not. So I think this is very important. I think lymphatic filariasis is one of the diseases where a lot of social science would help, because also the mass drug administration programs, they have the difficulty that, of course, they bring down infection, but usually it's the ones that look completely healthy that have to take the drugs, because they have the parasites to transmit. And the ones with the sick legs have already overcome the parasitic loads, but at the expense of morbidity. So these ones you actually don't have to treat. And this is very difficult to explain without the biological knowledge to a community. You know, they will then say, okay, why can't you help these ones with the thick legs? And we have to say, okay, they are already too far into their disease, and they can do foot hygiene, they can wash their feet, they can change their job from rice growing back to other crops. And therefore, it would be so important that we have a drug like doxycycline that helps, because it would increase very much the morbidity programs that the WHO has to enroll now for their 2030 roadmap.
Thank you. Thank you very much. This was very clear. Okay, now we have, I don't know if you would like to see yourself in the chat. We have, I think, 3 questions from Alma.
Okay, yeah, I can read it if you want. So the first one is, what's your take on the varying performance of the countries in the 2012 initiative, which was evaluated in 2016? This was a table with a red and a brownish and a green category, I remember. Well, I mean, the WHO stance has not been sort of to criticize the countries who have not been able to perform that well. Maybe in some countries, yes. For example, there are rich countries like Gabon. There's a lot of money coming in through oil, but the country's elite is just not really interested in its broad majority in NTD control, because it's sort of a side business, and they haven't even started to do some of the control activities. Of course, there is gentle diplomacy working, and I think the take now is digitalization. So there is a program which I have shortly mentioned. This is ESPEN. This is called Expanded Special Program for the Elimination of NTDs. And they're sitting, there's a sub-branch of WHO, they're sitting in Brazzaville, Republic of Congo. And the main thing they are doing currently, in addition to giving advisory functions to the countries, is setting up an electronic database. So you can go to their website and look into the performance map of each country. And of course, this creates then the, so to say, to the competitiveness among the countries that I think is also a necessary tool, that, you know, countries with a similar general income of their population should also do in a similar way. And of course, there are some better performers and worse performers. Some is due to the fact that the well performers have seen additional funding. For example, big community trials, countrywide trials organized by the Gates Foundation. And of course, this hasn't made an impact. And others have not been able to do so for various reasons. One, I think the major one is, of course, civil unrest and internal problems of their health systems. And I think the German take also in this respect is to try to get the countries with the lowest income and the weakest health systems, bring them up. And the buzzword here is health system strengthening. And this is, for example, a big program by our Ministry for Mutual Development, the BMZ, which is also here in Bonn, that they do health system strengthening work, cross-sectional. So with the idea, if the health system is able to pick up more people with regard to universal health access, see the SDGs, then it will eventually also do better with all the control programs. So the question 2, where does One Health play a role in my research, in your research, as in our research at the Institute? This, I have to say, is not at the focus, because the filariasis infections are not zoonosis, except for maybe filariasis that is also present in the US and Europe and is in cats and dogs and rodents. But here in humans are usually the only hosts. And so they are definitely not zoonosis. Of course, the One Health concept comes in when it is when it comes to climate change, when it comes to hygiene. We have spoken about washing the legs, better resourcing of water taps and so on. Of course, this is something that will also become part of the WHO programs. I think this is a major difference between the old roadmap and the new roadmap, that there is much more cross-sectional activities. And there is WASH, for example, is mentioned, One Health is mentioned. So I think this comes in, particularly in my research at drugs. It is of course not a major focus because we don't work epidemiologically. And question 3, looking at your long and elaborate testing procedure and phases, what is your take on the rapidly evolved COVID-19 vaccine? Well, so there is no, well, of course, the good thing is the long year studies have not really suffered because we were always allowed, we had to apply for with the country's regulatory offices that we continue our trials. This was not an automatism because Ghana, for example, had a complete shutdown in the beginning, and we had to argue that it was against the rights of our study patients, or it was in the right of our study patients to get a proper follow-up, and we had to develop a hygiene concept in order to be able to see the patients at the follow-ups. So with that regard, COVID impacts could be avoided. We are not taking part currently in a vaccine trial because this is just organized differently. There is a big treatment trial also done by DNDi, I think within 18 countries on multiple treatment arms, where I sort of serve as a more or less unofficial advisor on what treatment arms to take. So I'm not completely taking my back to COVID. I try to help where I can. And the other thing is, because of our presence in the countries, we have, of course, also educated the people on how to protect themselves. And we are now in the final steps of a study trying to find out how prevalent has COVID been at all in rural areas in Ghana. The good news is that it has not been very prevalent, at least as far as serology speaks. It's almost nothing. And this also accounts for the fact that in rural areas, of course, social distancing can work better than if you live in a big mega city with a lot of fully packed buses packed with people, where you obviously expose yourself much more. I hope I have answered these questions in the way Alma van der Venius wanted.
Okay, thank you very much. Yeah, Alma is very satisfied with your answer. I see here, and as we are all of us. So thank you very much. We have already passed the hour. So thank you very much, Professor Horao. Thank you very much, Dr. Bromer, for this very clear account of your work on the transdisciplinary research area, life and health. I say it like formally, it's name. So, and thanks to all of us who participated in this colloquium. We hope to see you on screen again next week. No, thank you very much.
Yeah, thank you. It was a nice event, and see you soon.
See you. Bye-bye.
Thank you. Bye-bye.
Thank you.
Bye-bye.